Okay, everyone, we're good to go because we have those joining us online now linked into the meeting. So my name's Professor Grant McArthur, Executive Director of the VCCC Alliance, and on behalf of the Doherty Institute and VCCC Alliance, I'd like to welcome everyone today to, I must admit, what I've been looking forward to as a really exciting seminar for some time. May I begin by acknowledging the traditional owners of the lands at which we meet today, including the traditional owners of the lands of those joining us online, and I wish to pay my respects to elders past, present and emerging of the Wurundjeri people of the Kulin Nation, whose traditional lands we're gathering today. Also wish to pay my respects to all First Nations people joining us today in person and online, and thank you for your contributions to our nation and to our nation's health. So today we're joining to talk about what is a paradigm shift, and I haven't actually discussed this with the next speaker. I'll be introducing Professor Sharon Lewin, but Sharon and I like to have a little to and fro about the biggest breakthroughs that have occurred in our careers. And Sharon, of course, says, you know, combined small molecule therapies for HIV that have been truly astounding. And I say immune checkpoint blockade for cancer, and so we have a nice little to and fro about that. So I put it to you, could this be a third? Could this be a third? So just to set the scene there and get you thinking about that. Some housekeeping. The webinar's been recorded, and you can share it with your colleagues, can look at it online at a later date. So we have two keynote speakers and a panel discussion will be the structure today. Hope to squeeze in a question or two from the audience, but apologies ahead of time if we don't get that fitting in. Sharon will be making some opening remarks shortly, and then we'll be hearing from our two keynote speakers, Paul Burton from Moderna and Shireen Loy from the Peter Mac and University of Melbourne. So without further ado, I'll hand over to Sharon for some opening remarks. Thanks, Sharon.
Thanks, Grant. I don't think it's really a competition between what has been more successful or how many more lives have been saved. I think COVID-19 vaccines are going to win out now. We haven't had the debate for a while. Grant didn't mention that we've known each other since a very long time. He was a year ahead of me at university, so just make that very clear. So since the mid-80s, we were residents together, and it's wonderful that we're working so closely together now across the road. Well, welcome everyone to the Doherty Institute. Special welcome. There's many familiar faces in the audience, but I wanted to make a special welcome to Dr. Amanda Caples, Chief Scientist of Victoria, and of course, Dr. Paul Burton, who's the Chief Medical Officer of Moderna. Fantastic to meet you, Paul. I was just hearing that Paul's partner and wife is from Melbourne, which means that Moderna is even more heavily invested in Melbourne than we really even knew. So that was good to hear. And for those that haven't been at the Doherty before, we're a joint venture of the University of Melbourne and Royal Melbourne Hospital. We work entirely on infectious diseases and immunology through research, education, and public health. And I think I can speak on behalf of all of our scientists and clinicians here at the Doherty that we, and actually really every person on the planet, has been blown away by what mRNA delivered in our response to COVID. We never really imagined that we would have a vaccine, an mRNA vaccine, so effective and also so widely distributed across the planet. But I think we're even more excited about the future potential of mRNA, not just to prevent, but also to treat infectious diseases and, of course, cancer that we're hearing about today. So Those 4 letters that I love saying, even a taxi driver knows them. Not that there's anything wrong about taxi drivers, but mRNA, everyone knows what those letters actually mean. And they have really captured the imagination of so many scientists around the world, but already here at the Doherty. So we've got groups working on mRNA vaccines for malaria, for HTLV1, for norovirus, and already showing really some fascinating results. And we're going to see a lot more of what mRNA can do for preventing infectious diseases. But what's really exciting to me about today's meeting is the potential for mRNA as a therapeutic. And we're going to hear about a super interesting study of an mRNA-tailored cancer vaccine given together with anti-PD-1 that led to significant success in treating melanoma. Now, this is just groundbreaking on so many fronts, as Grant already alluded to. So first, the use of mRNA as a therapeutic vaccine. Now, in infectious diseases, and particularly chronic viral infections such as HIV and hepatitis B, as well as cancer, as we were just discussing, the field is littered with many failed therapeutic vaccines. Most of us have thought they are never going to work in either chronic viral infection or cancer. So perhaps mRNA is really going to change that whole field, something we never thought was possible. The second, for me, is the combination of an mRNA vaccine with anti-PD-1, and what that is not only going to tell us about new therapeutics, but teach us about fundamental immunology and how to really enhance a vaccine response. And combining a vaccine with anti-PD-1 or actually other immune checkpoint blockers like anti-CTLA-4 has been done before for other cancers, but actually with very mixed results. And I've personally got a really strong interest in this strategy as and Grant knows again about this. I've been very interested in the role of anti-PD-1 as an HIV cure. And together with James McMahon at the Alfred, we're running the first clinical trial of anti-PD-1 in people with HIV on treatment who don't have cancer. And when we were designing this trial, we looked really closely at whether we could combine it with a vaccine as well, and there was nothing really suitable. But these studies of anti-PD-1 and actually mRNA for HIV, an mRNA vaccine for HIV, are actually now happening in animal models and also looking really promising. So what is going on here between this magic combination of anti-PD-1 and mRNA? And finally, we're hearing about mRNA as a therapeutic. And here the potential is just immense. And many scientists, again, across the Doherty have been exploring this, not through therapeutic vaccination, but through the delivery of nucleic acid targeting antivirals, such as Cas13 for SARS-CoV-2, HIV, hepatitis B, using mRNA to deliver those components of gene editing, and using mRNA to also deliver broadly neutralizing antibodies. And we think that this technology is going to be really important for therapeutics, and it will be a major platform of the new $325 million centre that we launched here, the coming Global Centre for Pandemic Therapeutics. But finally, I'd just like to reflect for a moment of where we are all sitting right now. Now, of course, we're all sitting in the Doherty, but we're also sitting in the largest biomedical precinct in the country, in a city that prides itself on being the medical research capital of Australia, and in a state that has put its money where its mouth is and invested heavily in providing the infrastructure we all need to do our work. So just look at the Doherty, built in 2014, VCCC in 2015, the Australian Institute for Infectious Disease, a new $650 million facility that will be a joint venture of the Doherty, the Burnet Institute and University of Melbourne, which will open in 2027. I mean, these are just extraordinary facilities and an incredible place to work. And the Victorian government also rapidly saw the power and importance of mRNA science. I remember those discussions very, very well with the Treasurer, Tim Pallas, at the time. And the establishment of mRNA Victoria in 2020, with an initial investment of $150 million, meant that many researchers across Victoria, including many here at the Doherty, many at Peter Mac, were able to jump into this new and highly competitive field. And I think that also meant that Victoria was able to attract Moderna. It wasn't just Paul's wife that attracted Moderna to establish its Asia Pacific headquarters in Melbourne, but it's really now an incredible ecosystem. So I'm super excited to hear today's talks, but also look forward to the incredible advances that mRNA, our brilliant Victorian scientists and clinicians and industry partners are going to deliver in the years ahead. So thanks very much and welcome again.
Thanks very much, Sharon, for that brilliant overview of the field, but also, like Sharon, I share the incredible excitement about working in this precinct and where Victoria and Melbourne sits, globally in biomedical research. So I could wax lyrical about Sharon's CV, but just the context of, where Victoria and Melbourne, where biomedical research sits. Sharon in 2014 was Melburnian of the Year, a scientist, a clinician scientist, and furthermore in 2020 won the Melbourne Achiever Award. Paul, I think that signifies where, this area of activity sits in our state, and we're very incredibly excited at Moderna for basing their headquarters here for the region in Melbourne. So now I'd like to kick off with the interesting stuff, which is the presentations today and the details, and so introduce the first keynote speaker, Paul Burton. So Paul is Chief Medical Officer at Moderna Therapeutics. He's had that job since 2021. And what's great, I think, about Paul's job, he's got global responsibility for all of medical strategy. So just put in place what Sharon just summarised about mRNA. Taxi drivers know about it. So, Paul, you've probably got the coolest job in the world, I would imagine. So, Paul got a PhD University of London and did train in cardiothoracic surgery. So, Paul knows cancer as a result, given the huge burden that thoracic cancers have globally. So, Paul, thank you so much for travelling all the way to Melbourne, and really look forward to hearing about from COVID to cancer and mRNA technology. Thanks, Paul.
Great. Well, look, thank you so much, Professor McArthur, Professor Lewin. It's a real honour to be here today, and we are thrilled and delighted as Moderna to be in this amazing ecosystem. As Grant said, I arrived yesterday. I've had some amazing meetings, and just the vibrancy and the as you said, the fact that this really appears to be such an amazing epicenter of clinical research and science is palpable, and it's been amazing. I want to thank the Doherty and the VCCC for this chance to talk about this today. So, look, I have two parts to this presentation. One that I'll go through fairly quickly, which is to talk about COVID a little bit, the vaccine, and where we've come in terms of protection against respiratory pathogens, and then to get into cancer and to talk about that. So this is the central dogma of biology, that DNA goes to RNA and goes to protein. You all know this far better than I do. It's interesting to me, though, that really this came out of a tea-time lecture that Francis Crick gave in 1957. So you can imagine Crick makes his own slides, probably types them out, puts them up on an acetate. It takes about a year for the notion of the central dogma of biology to gain ground. Imagine today we would be tweeting and Instagramming and talking about it, and by Friday morning it would be set in stone. But it took a year for it to take off. The remarkable thing, not only on that, is that Crick didn't know that mRNA even existed. right? He imagined that there was, he knew that there was DNA, he knew that there were proteins, and he hypothesized that there was this intermediary molecule. I say that because mRNA has clearly been around for a long time. Moderna was formed in 2010 and has spent the last decade and a bit perfecting not only the production and the fine-tuning of messenger RNA, how to make it, how to optimize it, but really to work very, very diligently on the lipid nanoparticle. And it's that lipid nanoparticle, or LNP, that's so critical in terms of uptake of messenger RNA, tissue-specific distribution of it. And the amazing thing about mRNA is that it will do exactly what your cell intends it to do. It can make a protein that will remain within the cell, gets translocated to the cell membrane, translocated to the nucleus, wherever it needs to go, or secreted. So remarkably powerful. We take messenger RNA and we put it in a lipid nanoparticle. We spent 10 years, as I say, really developing the constituents and the components of that lipid nanoparticle. It's a gentle lipid nanoparticle. It's biodegraded. It's broken down relatively quickly, what isn't taken up. And that's important because typically the amount of messenger RNA to lipid nanoparticle that is delivered is in a 1-to-1 ratio. So if you have a tolerable lipid nanoparticle, you can dose mRNA high. For our primary vaccination against COVID, we used to give 100 micrograms of messenger RNA. We got a profound immune response, and that translated into great clinical protection against infection, severe disease, and death. With the cancer therapeutic vaccine that I'll talk about in a minute, we can give up to 1 milligram of messenger RNA. What this cartoon also shows you is that within that lipid nanoparticle, you can bring multiple different messenger RNAs together. Right now, the COVID booster that's available here in this country and around the world has 2 different messenger RNAs. That presents 2 different spike proteins to the immune system, unlocks the power, the epitope broadening of your immune system. We're making a vaccine against cytomegalovirus, the leading cause of birth defects in newborns. It's a complex virus. We bring together 6 different messenger RNAs. So we can put multiple mRNAs into a single lipid nanoparticle. The other power of mRNA is the speed. We learned this from the pandemic, but typically, and Professor Lewin will talk to this perhaps, it takes between 8 and 12 years to make a vaccine by the time you do the preclinical work, toxicology, and then to test it. We actually went last year from the recommendation for a new COVID booster being based on the BA.4/5 strain of Omicron to manufacturing hundreds of millions of doses within 12 weeks. That was a new vaccine. Yes, we had perfected the technology, and yes, we had rolled out a COVID vaccine for the pandemic, but we really went from being told what was to be in a new vaccine to designing, producing it, and then scaling up manufacturing within just a matter of weeks. The RSV vaccine here that you see on the bottom of this slide, we've released data in recent weeks on that, 84% vaccine effectiveness with that vaccine in a 34,000 person study. We designed that and started testing it in 2021. So it's May of 2023. We were able to go from design to production and testing in a very large randomized controlled trial within 2 years. Again, that just speaks to the power of messenger RNA. And that's shown here, speed of production, but also the ability to take, as I said, multiple different messenger RNAs to tailor the design of a vaccine, either to a circulating strain. Imagine a world where within a flu season, you could adapt your vaccine and go out with a new vaccine. That world is possible with the messenger RNA, whereas potentially it would not have been possible with some of the other technology. So we're in respiratory vaccines, we're in latent viruses. We're also beginning now work in bacterial pathogens, Lyme disease, for example, an important medical condition, but also underpins the possibility of going after bacterial targets, which might overcome evolving, growing antimicrobial research. And we're working in global public health as well. We're designing programs up against the 15 priority pathogens, tuberculosis, malaria, and other important diseases worldwide. But we're more than a vaccine company. We're in personalized cancer therapeutics. I'll talk about that in a moment. We're in rare diseases where we're able to target enzymes that are deficient within mitochondria. Again, mRNA is so powerful there because the protein is produced, targeted to the organelle, the part of the cell where it needs to go, fulfills its role. We're doing a collaboration with the biotechnology company Vertex. We have an aerosolized and inhaled lipid nanoparticle that's able to combine a messenger RNA to combat cystic fibrosis. Or even in cardiovascular disease. We have programs in heart failure. We've even encoded messenger RNA. We're able to inject into the heart during bypass surgery to see if it can induce neoangiogenesis of blood vessels in people with cardiovascular disease. So let me talk a little bit about cancer and our individualized neoantigen therapy, or INT. We've heard a little bit about the search over decades for a vaccine-directed immunotherapy for cancer. And what we do is shown here in this cartoon. We take some cancer tissue, we take some normal tissue, we do deep sequencing to identify those neoantigens, novel antigens that are specific to the cancer cell that may drive cell biology of that cancer cell, division, metastasis, prevention of apoptosis. But we identify those neoantigens, and we're able to identify within a protein the core area of that protein that would be most immunogenic. And what is remarkable to me is that we can then stitch together up to 34 different messenger RNAs, so targeting 34 different neoantigens, and put that mRNA into a lipid nanoparticle. We then inject it into the deltoid, into the arm muscle, just as we do when we get a vaccine for COVID. As I said, we give about 1 milligram of that therapeutic, so 20 times the current booster dose that we get with COVID. We do that every 3 weeks for about 9 months. We immunize people repeatedly, and their immune system takes the messenger RNA, presents and decorates those proteins on antigen-presenting cells, presents that to our immune system, and then really allows our immune system to do the rest, to unleash the power of T cells, maybe B cells as well, to turn on the cancer. So we have done a study in melanoma. Melanoma is interesting because it has a high degree of mutational burden. There are a lot of neoantigens. You can see that here in this graph. Compared to other types of cancer, it has a disproportional representation of mutational burden. Melanoma, we know, remains an important unmet medical need. Here in Australia, clearly melanoma prevalent, overrepresented in terms of the spectrum of different cancer that patients present with. Maybe 16,000 patients a year here will present with melanoma of different stages. And the survival rate for those more severe patients is still very low. I did my surgical training in the 1990s, and for a time I was a surgical house officer at the Marsden Hospital in London and did a melanoma job there. And the survival for patients, even with radical dissection and lymph node dissection, for those with severe disease was poor, and it has not improved substantially even today. So we did a study which is shown here. We randomized people 2-to-1 to receive either Keytruda, one of the most effective immunotherapies for melanoma today. These were patients with stage 3 and 4 melanoma, which represents about 5%. So in this country, it's about 750 people. And Australia was a major contributor to enrolling into this study. So these patients had stage 3 or 4 melanoma. They'd been surgically resected, so they were macroscopically clear of disease, and they were then randomized to receive Keytruda alone or Keytruda plus an individualized neoantigen therapy, specifically handmade, each one of these handmade against their cancer. We randomized them, immunized them every 3 weeks, about 9 months, and then followed them over a period of time. Now, this study actually started in 2017, 2018. So even before the pandemic. It took a bit of a slowdown during the pandemic, and then we were able to ramp up production again, really in 2021 and onwards. But it took a long time. As I say, each one of these is handmade. It's hugely labor intensive. It takes about 8 to 10 weeks from the time we call needle to needle, from getting the biopsy to getting ready for the first immunization into a patient. So it's exquisitely labor intensive, but it's extremely ultra personalized and patient specific. And what did we find? Well, we unblinded the study. We presented the data at the American Association for Cancer Research, the AACR meeting a couple of weeks ago. We talked about the top line results some months ago, but we presented the full data set a few weeks ago. And this is what we saw. Here's the Kaplan-Meier estimates of effect. The black line is for patients who received Keytruda alone. Interestingly, the 1-year survival, progression-free survival rate at that time for the Keytruda patients alone is 77%. If you look at the data from the Keynote 054 study, which really served as the foundation for approval of Keytruda in stage 3 melanoma patients, the 1-year survival was about 75%. So at least some kind of internal validation that the patients and the therapy in that control arm was behaving as you would expect it to. You can see the patients who then received Keytruda plus their individualized neoantigen therapy in red. There's some improvement at 1 year, 83.5% progression-free survival. But after that, you can see the curves begin to separate. By 18 months, the rates of progression-free survival are 78 and 62%. So overall, a 44% reduction in the risk of a patient's melanoma recurring or dying from their melanoma, 44% reduction compared to Keytruda alone for those people who receive Keytruda plus their individualized neoantigen therapy. What is the safety profile like? Well, we're so used to seeing new additive chemotherapy regimens come along, which present to patients an opportunity, but also an increased additive, often risk of adverse events. I think very reassuring here that the rates of adverse events and important, more severe adverse events, grade 3 or more, really very similar between the two groups. So the therapy, even at 1 milligram given every 3 weeks for 9 months, did not appear to increase the adverse event profile compared to Keytruda. So, in summary, I think we're at a crossroads, a turning point, potentially in the management of melanoma, potentially in the management of cancer, where we can personalize to a very high degree, up to 34 different neoantigens tailored for somebody's individual cancer. We can put that into a lipid nanoparticle, we can repeatedly administer it to patients, and we can ignite somebody's immune system to present a T-cell response, perhaps a B-cell response as well to their cancer. Appears to be effective, appears to be well tolerated, and we hope that results of this study could be the basis of an initial approval for the management of melanoma patients. And over time, we're now beginning again with our partners, Merck, to expand into melanoma again, but now into lung cancer, colorectal cancer, and even tumors that have low mutational burden, and see if we can improve the outcome there as well. We've received breakthrough designation and prime designation as well for this therapy. So thank you for your time.
Thanks very much, Paul. Very exciting data indeed, and for emphasising the remarkable speed, isn't it? You have to almost pinch yourself to think about that time to development of those mRNA-based vaccines. So next keynote speaker is Professor Shireen Loy, who's head of Translational Breast Cancer Genomics and Therapeutics Lab at the Peter MacCallum Cancer Centre, and Shireen is also the National Breast Cancer Foundation endowed chair at the University of Melbourne. Shireen today will talk to us about the clinical implications of these mRNA technologies, really building on her expertise as one of the leading immunotherapeutic breast cancer researchers in the world. Because I think the exciting thing is to think beyond skin cancer and the high neoantigen load cancers to implications for other tumours. So I look forward, Shireen, to your thoughts about where we're going. Thanks, Shireen.
Good afternoon, everyone. It's a great pleasure to be here, and thank you, Grant, for the invitation. So I'm a medical oncologist at Peter Mac, so I've been tasked today to provide a clinical and oncological perspective on the potential of the therapeutic abilities of such an approach. So I work with breast cancer, which is not like melanoma, which is the immunotherapy poster child. So we've learned a lot of things trying to develop immunotherapeutic approaches in breast cancer, and I think it's interesting to reflect on how we can bring that forward with this particular new therapeutic strategy. So these are my disclosures. I've noted I have received no funding from Moderna. Not yet. And next slide. Okay, why can't I go forward? Okay, nope. Okay, all right. So as our previous speakers have so eloquently alluded to, the COVID pandemic has really put mRNA technology on the global stage, and really it's amazing to reflect on how global efforts have really accelerated our response to the pandemic, and also to the technology, the optimization of the technology, structure, stability, and delivery of the technology. But prior to the pandemic, of course, COVID mRNA vaccines were being explored as a cancer therapeutic strategy. And this is because, for a number of reasons, they are very effective at producing an immune response, both humoral and cell-mediated. They have low toxicity compared to other cancer strategies. They're relatively fast and inexpensive to manufacture. They're easily degraded. They're not incorporated into the host genome, and potentially they're non-infectious as well. So now we have proof of concept that this approach is effective in a solid cancer type, as you've just seen from Dr. Burton's presentation. I think this trial provides for the cancer field a number of proof of concepts that a vaccine can actually work in cancer patients. So as previously alluded, we've had lots of different vaccine approaches which have never gone anywhere fast. The mRNA vaccine approach can work in cancer. The neoantigen individualized approach can work in cancer patients, though admittedly this is a melanoma population which has high degree of potential neoantigens. The algorithms that predict the neoantigens specific to the patient's HLA type seem to work as well, which again is proof of concept. This treatment is acceptable and well tolerated by the patient. We know in cancer, treatment compliance is a big issue, particularly in the early stage settings. So quality of life is very important. And logistically, such an approach is possible as well, because cancer patients have far more degree of urgency for their treatment to be given in the early stage setting. So the fact that this labor-intensive approach can be rolled out in this particular population for many patients, and is, again, proof of concept that we can expand this to a much larger number of patients to other tumor types. This is a randomized phase 2 study. The advantage of the randomization is that we can compare against standard of care, and that's really important in going forward in cancer therapeutics, because then the next trial is a much larger phase 3 study where we're comparing against standard of care, which are currently approved by the Australian government Medicare system. So again, first individualized neoantigen approach to be efficacious in a cancer setting. So what have we learned since we've been developing immunotherapy in cancer patients, and particularly in cancers such as breast cancer? So it matters who you include in these trials. So traditionally for drug development, we've always looked at the advanced cancer population. These patients have often had many lines of treatment. And we've learned, the hard way, particularly in breast cancer, that this group is not the best group to test any sort of immunotherapy approach. The reasons for this are multiple, but their immune systems are likely dysfunctional due to many lines of prior treatment. They have large cancer burden, and the actual tumor microenvironment is also immunosuppressive, which is not the best environment for immune cells to infiltrate and be efficacious. I think recurrent disease, or disease that has relapsed, is most likely immune deficient or immune resistant by definition. And it's not a surprise, as we know in the infectious community, that you need your vaccine prior to exposure. This is the case for flu vaccine, for example. So as examples of these vaccine approaches have been tested in the advanced setting. And whilst the advanced stage is good for looking at potential toxicity, it's really hard to interpret any sort of efficacy, because these patients are usually heterogeneous in their prior treatments, their cancer burden, and also any sort of T cell or immune response. So this is a similar vaccine that's just been presented as successful, but really the early studies did not really give much of a hint that it would be efficacious. This mRNA vaccine in gastrointestinal cancer in the advanced setting, again, no responses were seen, clinical responses. And this mRNA vaccine developed by biotech, which is a different type of antigen, again, in the advanced setting, showed some evidence of T cell responses, but again, very little evidence of objective clinical responses. So in the advanced setting, we have large cancer burden. We have many clones which are capable of resisting treatment. And also we're beginning to learn that the cancer cell and cancer ecosystem is very plastic in that it's dynamic and can change according to treatment and, of course, to the host response. So in cancer, we've certainly had to move towards strategies of combination therapy as well as sequential therapy. And we know that we are more likely to cure the patient if they have minimal residual disease or no macroscopic disease present. For example, in breast cancer, we use chemotherapy routinely, and this is because it removes the majority of the cancer cells or cancer clones. But we know that we need to follow this up in most patients with long durations of maintenance therapy. So we follow this up with endocrine therapy, 5 to 10 years often, Herceptin or HER2 targeted therapies for 1 to 2 years, PARP inhibitors, CDK4 inhibitors. And so these therapies are maintained for many, many years in order for us to cure the patient. And we believe that's because they can effectively target these remaining 1% of clones which are dormant or resistant to the previous therapy, and reflects the cancer cell's ability to change and evolve with the treatment that it's been given. So the adjuvant setting where there's no detectable disease is most likely better for these immunotherapy approaches. We've seen proof of concept of that. But the most important thing is that it needs to have a good toxicity profile, because for the patient, they're getting back to work and their normal lives, and also that the treatment needs to be acceptable to patients. So in breast cancer, these patients have extremely high rates of non-compliance with their endocrine therapy. We know that, and we know that also impacts on survival. So this data has already been presented. We can see that this treatment is very efficacious. I think it's interesting to perhaps think about what's happening up here before the curve splits, so into the first year. Is that because of it takes that long to generate a new immune response? So that I think is quite interesting to perhaps understand a little bit further. I think the most interesting data for us in the cancer field is these particular curves, which look at the efficacy according to PD-L1 status. So PD-L1 is kind of our surrogate for a pre-existing immune response. And there's a lot of talk in the cancer field of turning cold tumors into hot tumors, etc. We haven't really been able to do that successfully, particularly for the non-immunogenic tumor types such as breast cancer. But you can see here that in this particular group, the patients on the control arm that were PD-L1 negative doing extremely poorly compared to patients receiving the investigational agent and pembrolizumab, who are doing much better in comparison. So this suggests that this combination can effectively induce a new, durable, and robust immune response that can be enhanced with the addition of pembrolizumab. Excuse me. I do not have COVID. Okay, so as mentioned, excuse me, this treatment is well tolerated, which is extremely important for compliance. So how do we move forward, and what is the right phase 3 trial design, and which patients do we choose for such a therapeutic strategy? So as we've already alluded to, the patients who are high risk, so most likely to recur on standard therapy, excuse me, they must of course have tissue available because we have to sequence them to identify potential antigens. Given recent data, it seems very clear to me now that the standard approach will be neoadjuvant immunotherapy and standard of care. In some cancer types, this will be combination with chemotherapy. But of course, this gives us enough time to generate what's required for vaccine production. How many neoantigens will we include in such a vaccine for other solid tumor types? And for tumor types that have a low mutational burden, how are we going to choose the particular antigens to include there? And then, of course, after we give this pre-surgical treatment, which patients are we going to give this approach to? So often patients have either a very good pathological response after surgery, and some patients have a very poor pathological response at surgery. This is often correlated with prognosis. So in breast cancer, if you achieve a complete pathological response, so no invasive tumor at surgery, your prognosis is excellent. But it also suggests that you've had also probably the best immunological response to your treatment. So perhaps these patients, even though they do very well, are also very good candidates for immunological approach. Some patients have residual disease, but this disease is filled with lymphocytes or immune cells, potentially suggesting that these patients are amenable to further immune approaches. But then we have some patients who have marked residual disease, and these patients are most likely to have both chemo and immune refractory disease, and perhaps these patients need to be removed out of the pool for future immunotherapy approaches. There's a lot of talk about ctDNA or minimal residual disease now as a marker for these approaches. I personally think that this may also reflect a disease state which is very aggressive and again may not be susceptible to an immune approach. This of course needs to be further validated, but certainly in breast cancer, if you are detecting ctDNA at surgery, you're doing extremely poorly, and it doesn't seem that we're able to change the natural history of those patients at present. So these patients could be randomized to the combination of the individualized vaccine and a PD-1, PD-L1 inhibitor. As well as you can also consider patients who achieve a complete pathological response. And of course, how are we going to monitor these patients? It'd be good if we can have some sort of short-term surrogate endpoint which reflects long-term outcome, because in clinical trials, we're often waiting a very long time for the final endpoint to read out. So often we're looking for short-term surrogate endpoints which can help us identify if the approach is going to work. And so finally, just potentially one last approach for this type of strategy is prevention of high-risk individuals. So an example of this is this small study which looked at a MUC1 peptide vaccine for prevention of recurrent colorectal adenoma. This was given to patients who had high risk of developing adenomas. So these patients had previously had adenomas and adenomatous polyps. They received a MUC1 peptide vaccine or placebo. And you can see there not everyone had a good immune response. Around 25% of patients had a good immune response to their vaccine compared with placebo. But importantly, in those patients who had the immune response, they had a 38% reduction in adenoma recurrence compared with those patients who received placebo. So this is in the prevention high-risk setting. So how do we go forward potentially with this type of approach? We need to select high-risk individuals, for example, those at high risk of colorectal cancer. In breast cancer, patients with germline BRCA1, 2, or PALB2 carriers are the most obvious group to go for. P53 germline alterations, and of course, we can consider the smokers who are at high risk of non-small cell lung cancer. I think the most important question here, of course, is what antigen are we going to include in the vaccine? There are a number of different approaches to this that we could consider. And the trial could be vaccine versus placebo or vaccine plus a PD-1 inhibitor. We may not need a year of that. And again, short-term endpoints here could be highly relevant, imaging or colonoscopy. Toxicity is likely to be low, and therefore we accept that this would be acceptable to the patients. We know that in breast cancer, patients often refuse to take their chemoprevention because of the side effects. But for this particular strategy, very large trials are needed and will require serious investment and long-term follow-up required. But of course has the highest gain with regards to reduction of cancer burden and mortality and morbidity globally. So in conclusion, this particular field is moving rapidly. It's going to be game-changing, we feel. The phase 3 trials are starting or ongoing. Of course, our ability to upscale, manufacture, produce these vaccines are going to be critical, as well as the regulatory hurdles that will accompany that. Patient selection is likely to be critical. And as I mentioned, the low toxicity is going to be an advantage. Combination strategies in cancer are likely to be key. Many technical details to work out. But obviously, in the prevention context, there's high risk but high gain. So thank you.
Thanks, Shireen, for giving us insight into the future. If I could get our panel members, please, to come and join us up the front. And I'll, as people are taking their seats for the panel discussion, I'll start some introductions. So we're really delighted today to be joined by Dr. Amanda Capels, who is Victoria's lead scientist and a director of Breakthrough Victoria. You know, I mentioned that maybe Paul's got the coolest job in the world, but Amanda, here in Victoria, you've got to have, you know, at least the second coolest job in the world, being in this environment and leading us in our scientific endeavours. Also joining us is Dr. Karen Innes, who is a Consumer Reference Group member and Governance Committee member from Melanoma Research Victoria. And Karen and I actually did our PhDs at about the same time. And Karen trained in a PhD, actually in molecular immunology, and has since been working in more recent years as a patent attorney, but joins us today because of her lived and living experience of receiving immunotherapy for melanoma. So fantastic to have you here today, Karen. And last but not least is Professor Laura Mackay. So Laura is lab head and the immunology theme leader here at the Doherty Institute. And amongst her amazing CV, the thing, Laura, that, you know, I have said to you that I think is completely cool is that in 2022 she was the youngest ever fellow elected to the Australian Academy of Health and Medical Sciences. So that that's a testament to the quality of her research. So I'll ask our panel members some questions, and if we get time, we'll try to fit in a question or two, you know, from the audience. So I might start with Amanda. So both Sharon and I, in the introduction, Amanda, you know, were expressed our excitement about being able to do our work here in Victoria. Why is it that companies like Moderna have invested in manufacturing facility and research investment in Victoria, BioNTech, the other main mRNA biotechnology company has got significant investment coming as well. Why Victoria? Why do you think Victoria is attractive for these world-leading biotechnology companies?
Thanks, Grant. And in part, I think Sharon has probably already addressed that question, because if you think about what Sharon said and what we know that exists here in Victoria, is that we've got a strong science base. We have two universities, the University of Melbourne and Monash University, that are ranked in the top 50 around the world. So if you want to establish an industry, if industry is attracted to where there is excellence in science. They're also attracted to a place where, and it goes hand in hand with doing excellence in science, where scientists have access to the tools and the technologies that they need to do that science, and in the infrastructure, the buildings that we're sitting in here today, that enable them to get on with it and do that job. So when the pandemic hit and when we saw the power of mRNA technology, we knew what needed to be done, and that was for a dedicated strategy to invest in good science, which we did with mRNA Victoria. So we established mRNA Victoria to invest in the sector, in the science with our acceleration grants, in the ecosystem with the activation grants. And then because of the speed with which we knew that we needed to respond to attract Moderna, and as you mentioned, also BioNTech to Victoria. So we knew what to do, and the state government has had that long-term commitment. We had the playbook because we've done this before over a longer time frame with the Victorian Cancer Agency. And the final thing that I'll say is that as policymakers, we know that we need access to the best information and expert advice. So the formation of the Scientific Advisory Group, of which Sharon is a key member, was a really important element to our overall strategy.
Great, thanks, thanks, Amanda. I might turn now to Laura. So, Laura, you know more about T cells than anyone in this room, I'm sure. But there's actually many subsets of T cells. And in cancer, we often focus on CD8 T cells as effector cells for killing cancer cells. But for these individualized neoantigen vaccines, mRNA vaccines, should they be designed to target more than those CD8 effector T cells?
Yeah, thanks, Grant. Yeah, it's an important issue because CD8s work in concert with a lot of other cells. And certainly, apart from unconventional T cells, there's always CD4 T cells also. And so you might look at the number of CD8 T cells that you're getting from a certain vaccine, but without CD4 T cell help, their functionality actually might be compromised. It's been shown that they need that. But also when you're thinking about the types of CD8 T cells that you boost within themselves, the CD8 T cell pool is immensely heterogeneous. And so you can get different flavors of T cells that have different properties to go different places and have different functions. And broadly speaking, you can have T cells that can go into tumors and, establish residence there and be highly protective. They've been recently correlated with cancer control and efficacy to respond to for patients who respond to checkpoint blockade. And also you can get T cells that you can gear to sort of go more to the circulation. And these T cells will be the ones that you're reading out when you look in the PBMC. So if you're looking at the efficiency of your vaccine and you just look in the PBMC, you might be missing the whole picture. But thinking about the type of response that you're gearing towards, those that are going to go into the tumors and be helpful, and maybe those that would be less helpful that might just be circulating around. These decisions are going to be massively impacted by the route of delivery, the delivery vehicle, the adjuvant, and the combination therapy that you're giving. And so this all needs to be taken into consideration. And there's definitely, I think it's very unlikely there would be a one-size-fits-all for a type of regime that would go between cancers and type of route of immunization that might be more or less successful between melanoma versus breast cancer, for example.
Great, thanks, Laura. I might turn to Karen now. Karen, you've received, what might be described as the sort of blunt or indiscriminate form of immunotherapy for cancer. What impact do you think, this new technology might be for patients with cancer.
Yeah, thanks, Grant. Yeah, in my opinion, I think this new therapy will have substantial benefits for patients. Including on issues of tolerability of treatment, response rates, and recurrence rates. I have a background in immunology and I work as a patent attorney. I draft a lot of patent applications in melanoma. And so I would read these statistics, with a sort of somewhat detached approach, to, complete response rates and things like that. But I must say, being a melanoma patient, it puts those statistics in a whole different light. And so things like, 44% reduction in risk of recurrence is very meaningful from the perspective of someone going through cancer treatment. As Grant mentioned, immunotherapy is brutal. I might say. Just to give you a bit of perspective of my background and my treatment, I was diagnosed with stage 3B melanoma back in August 2020, right in the middle of lockdowns during COVID. So that's my COVID experience, was going through cancer treatment. We had the surgery to remove the primary, but I did have a metastases into the lymph nodes several months later, which required further surgery to remove the lymph node basin on one side of my neck. And then during that time, I was also undergoing immunotherapy treatment. I wasn't on Keytruda. I was on nivolumab and ipilimumab, which is Opdivo Yervoy. And I was fortunate enough to get through all 4 cycles, but it's pretty rare that patients get through all 4 cycles, because generally what tends to happen is that you start to get immune-related side effects and severe inflammatory side effects. So for me, that resulted in grade 3 hepatitis and neuropathy in my hands and feet. And then, of course, that meant going on high-dose steroids, and then going on high-dose steroids comes with it a whole other bunch of side effects that you get taking high-dose steroids. Fortunately, the neuropathy in my hands has improved, but I'm probably left with the permanent legacy of neuropathy in my feet as a consequence of treatment. But, happy to say that, I've been clear for 18 months now, but still going through the lived experience because I'm still having PET scans every 3 months. So as I like to say to Grant, I tend to refer to this as the scanxiety, every 3 months of kind of bringing it home again of kind of what you've been through. But one of the other, going back to the point of these sort of targeted treatments, I think, the way I see these is it's a really bespoke approach to treatment for patients. And, in my personal opinion, I think this is where medicine's going. It's heading towards personalised treatment. And then hopefully with that, what it means for the patient is that you're starting with a therapy that is specific to the patient and specific to that patient's tumour profile. And then hopefully what that might mean is that you're not finding yourself starting on one type of treatment, finding that type of treatment's intolerable, and then having to go to another, or finding you're not responding to one type of treatment and then having to go on to another, and then you find yourself jumping from one sort of treatment to another. And I think this was touched on beautifully when you talked about quality of life, because it's an important issue when you're going through cancer treatment as well. How do you sort of maintain, a reasonable quality of life while you're going through treatment?
Great, thanks, Karen, for sharing that with us. And what's interesting, that treatment that Karen just described in such detail, the toxicities, we actually did clinical trials in the adjuvant setting and was not beneficial compared to the anti-PD-1 alone, and yet brought extra toxicity. And I just reinforce the fact that the individualised neoantigen vaccine study, no increase in grade 3 or severe toxicity, and yet such a striking therapeutic benefit. So thanks, Karen. Very powerful narrative there for us. Now we're going to need to wind up soon, so I might just ask both Paul and then Shireen for some final comments. Take your pick about what the future holds, Paul, before we close.
Maybe just thanks, Grant. 30 seconds. Look, I think it is an important, profound clinical benefit, but I think it opens up what will be decades of new research to come. It really opens up a new avenue for important clinical treatment of cancer. So I think we are on we're on the shoulders of giants, but we have so much opportunity ahead. And just to say again that the chance for Moderna to be part of this ecosystem here in Australia is an amazing opportunity for us. There's so much we can learn, and hopefully we'll bring mRNA to Australia as well, and do it here in Melbourne. So thank you.
And Shireen, maybe your number one prediction for the future in oncology of mRNA technology. What do you reckon? And we won't hold you to it, but of course, if you're right, we'll be quoting it for years. Yeah.
Prevention.
Yeah, that's a really, really interesting.
Enough?
And what struck me, Shireen, from your presentation and Paul's is you might think prediction's very hard, but that Moderna mRNA vaccine had 34, up to 34 neoantigens. And so when that I think really gives hope in that prevention as space that Shireen presented. So apologies, we won't have time for questions from the audience, but I just want to thank everyone that joined us online, here in person. And thank you so much to Paul and Shireen, our keynote speakers, and all the panel members for giving us an insight here of what is such an exciting area of development in medical science. It's been a huge pleasure for VCCC Alliance to partner with the Doherty Institute today. And for those that are staying for the media call, please remain in the room to facilitate questions. And for everyone else, have a great afternoon. I think the future is bright for many health conditions as a result of this new technology. Thank you very much for your attendance.